Hi guys, Miss Francis here. In this video, we're going to explore the genetics of bacteria. So bacteria cells divide by binary fission. They replicate by binary fission. They don't do mitosis. They don't have spindle fibers. They have a single origin of replication in which replication occurs in both directions, forming two replication forks. But essentially what bacteria are doing is creating clones of themselves. So if they're just creating clones of themselves, then how did evolution occur? I mean, after all, if we all came from a single aquatic prokaryote and all they're doing is cloning themselves, well, then how did we come about? So some might argue, well, there were mutations that created differences, but I'd argue that random mutations occur so infrequently that there must be some other mechanism that caused prokaryotes to evolve over time. And in fact, we call that mechanism horizontal transfer. So horizontal transfer in bacteria allows for genetic variation in prokaryotes, where there are three different mechanisms for this horizontal transfer. There's transduction, transformation, and conjugation. So transformation we've seen very recently. That's what we did in our lab. Conjugation we discussed when we compared prokaryotes and eukaryotes. However, transduction is a new mechanism for us. So let's talk about the type of horizontal transfer that we have most recently performed. Transformation is the alteration of a bacteria cell's genotype by the uptake of naked foreign DNA from the surrounding environment. And guys, this is what we did in our lab. We had a bacteria cell uptake foreign DNA, where within a plasmid, we used transformation techniques. We exposed it to calcium chloride and then performed a heat shock so that plasmid would uptake foreign DNA. In our case, it was ampicillin resistance and the green fluorescent protein. So this is transformation. That is one mechanism for horizontal transfer. This is a mechanism we haven't seen yet. Transduction. It occurs when a virus that infects bacteria, known as a phage, I know everybody else says phage, I say phage, potatoes, potatoes, tomatoes, tomatoes, whatever, carries bacterial genes from one host cell to another. So let's annotate this image to explore transduction. So here we go. The first step in transduction is that the phage injects its DNA. And now what that phage DNA does, what that phage enzymes do is degrade the host cell's DNA. In this case, our host cell is a bacteria cell. Then what happens is that host cell's DNA gets taken up or combines with the phage DNA. It gets incorporated into the capsid, actually, not necessarily the phage's DNA. But this is occurring inadvertently. The phage does not mean to carry with it the host's DNA. And now the phage with that donor DNA, the donor being the host cell, in this case it's a bacteria, is called a transducing phage. Now once that transducing phage goes on to infect another bacteria, it takes with it the DNA from the previous cell, from the previous bacteria, known as the donor. So this is how donor DNA, which in the case of a phage is a bacteria cell, so this is how bacterial DNA gets from one bacteria into the next bacteria. Conjugation is the third mechanism for horizontal gene transfer in bacteria. And remember, when we were comparing prokaryotes and eukaryotes, we learned that conjugation is the transfer of genetic material between two bacterial cells that are temporarily joined. They are temporarily joined by what's called a pilus. That's the singular for pili, where pili are all these little offshoots coming from the bacteria cell, and the plasmid is what gets exchanged. So the male donates its plasmid to the female. Here's a really nice image that summarizes the 3 different mechanisms of horizontal gene transfer. So this first mechanism, conjugation, where the plasmid where the bacteria cells that are connected by a pilus exchange DNA via a plasmid. The second mechanism of horizontal gene transfer that we discussed was transformation, where the plasmid takes up foreign DNA. And lastly, the new mechanism that we haven't seen before is called transduction, where we've got a phage DNA called the transducing phage that carries with it DNA from the previous host cell. So how does horizontal transfer appear in evolution? Well, if we look at our evolutionary tree, scientists have divided organisms into 3 domains: bacteria, archaea, and eukarya. We fall in the eukarya domain. That represents eukaryotes such as plants, animals, protists, and fungi. Bacteria, that would represent organisms such as E. coli. E. coli is usually the classic example of bacteria. Archaea bacteria are known as your extremists. These organisms, these prokaryotes, can survive in extreme conditions, in either extremely cold conditions or extremely hot conditions. It was Taq polymerase, which would fall under this domain, that enabled humans to do PCR, because this Taq polymerase is a DNA polymerase that can survive extreme warm temperatures. So what these horizontal connections represent is that horizontal transfer of DNA. And would you look right here? Do you see how plastids, mitochondria, we could add chloroplasts? This is evidence for the endosymbiotic theory. Now, in our next video, we'll be discussing how prokaryotes regulate their genes. We already discussed eukaryotic gene regulation, which is much more complex than prokaryotic gene regulation, but prokaryotes do have a mechanism for regulating their genes, and that's what we'll discuss in our next video.